guys, welcome to your cell transport video notes. So cell transport simply is how things move in and out of the cell membrane. So we've already learned about the structure of the cell membrane, so now we're going to learn how things use that structure to move in and out of the cell. So first of all, there are two types of transport. So there's passive transport and active transport. So passive does not require energy and active does. It's kind of the same thing if you think about, if we draw a little hill here, if you're riding your bike up a hill, right, going uphill requires energy, so that's active. Once you reach the top and you start going downhill, that becomes passive because you could just go down the hill and not use any energy whatsoever. So passive transport is simply materials moving from high to low concentration. They do not use energy. Something called Brownian movement is what's used, and this is just a random movement of particles due to kinetic energy. And there's three types that you need to be aware of. So there's diffusion, facilitated diffusion, and osmosis. And we're going to break down each one of those. So diffusion, this is the movement of particles. Key word here being particles, from high concentration to low concentration, or another term you might hear used is down the concentration gradient. And this moves things like lipids, oxygen, carbon dioxide. If you look at the picture down here, you'll see that it's showing those gases moving through the cell membrane. And the whole point of diffusion is to work towards equilibrium or homeostasis for that organism or that cell. And something called dynamic equilibrium is what happens when the material inside of the cell equals material outside of the cell. Diffusion is also really easy to think of when you think about if someone sprays something, whether that's Lysol or maybe a really strong perfume, you eventually smell it even though you initially didn't smell it when it was sprayed, because those molecules are diffusing through the room or through the area that you're at. So diffusion rates. There's a few things that can affect the rate of diffusion. So temperature is one. Higher temperature means faster movement, faster diffusion. Pressure, same thing. A higher pressure is a faster diffusion rate. Particle size. As you can imagine, smaller sized particles are going to diffuse faster than larger particles. The collision number. So all of those particles are colliding with each other. So the more they collide, the faster that diffusion rate. And finally, the diffusion medium. So solids, those are going to diffuse the slowest, while gases are going to diffuse the fastest. So if that thing is in a gas, it's going to diffuse much faster than if it was in a solid or a liquid. So what about those larger molecules? So we show certain gases and molecules moving through, but what happens to the bigger ones? Well, the bigger ones use something called facilitated diffusion. So this is the movement of materials across the membrane, but this time we're using proteins. And those proteins are usually going to change shape to move the molecules, very similar to those carrier proteins we've discussed. And the important thing to note, though, is that we're still using zero energy. There is no energy being used because we're still going down the concentration gradient. So osmosis. Osmosis is diffusion, but it's only of water. Once again, high to low concentration. And the best way to remember this is H2O. And osmosis starts with an O. And this rate can be controlled by the solute concentration. So the higher that concentration, the rate is going to be affected much more than if it was a low concentration. So there's also three types of solutions, and this is relating to osmosis. So you have isotonic, hypotonic, and hypertonic. Now, iso is equal. Hypo, I like to think of hippo. So hippos are big and plump and round, right? So when you get a hippo or hypo, that also means it's going to be a large cell. The cell is swelling. Hyper, think about skinny. Someone that's really hyper is probably going to be pretty skinny because they're always running around and doing everything and they can never sit still. So someone that's hyper or hypertonic is going to be a cell that shrivels up. It's a skinny cell. So the isotonic solution. So this is when the solution has an equal solute concentration to the cell. So instead of, typically people like to think that there's no movement at all, there is still movement, it's just moving both ways. So our arrows would go in and out of the cell. There's still movement, it's just happening at the same rate. Hypotonic is when the solute concentration is higher inside of the cell and the solution has a lower concentration outside. So this is where we talked about the hippo. So hypo, remember, hippo, and this is when the cell is swelling. Eventually, animal cells here are going to lyse or explode, and this is ironically the ideal condition for plant cells. Plant cells want to be large because it helps them maintain their structure. So when they start to shrivel or start to lose water, they're going to also affect that structure of the plant. So a hypertonic solution, on the other hand, is when the solute concentration is higher outside of the cell. So in this situation, water's leaving the cell, and eventually the cell is going to shrink. This is also why plants wilt. So when plants are wilting, it's because they don't have enough water in their cells. And ironically, though, the cell structure stays the same. As you can see, all this like blank space in here, if you can see that, that is the cytoplasm in the cell membrane moving away from the cell wall. But because plants have that cell wall doesn't change, just what's inside of the cell does. So there's also a term called osmoregulation that you need to be familiar with. And we'll talk about this more when we get to fish, or if you ever take a zoology class or a class in college. But animals have to maintain that water balance. I should say plants too. And the whole point of this is to maintain homeostasis. And how they do this is they change their osmotic pressures within their cells. So why do I have a picture of fish on here? Well, think about it. Some fish live in fresh water, which is fine. They live in fresh water. It's very easy to maintain their water balance. But what about the fish that live in salt water? So fish that live in salt water have to regulate their water much more within their body, because otherwise it's going to affect them in a very negative way because of that salty water environment. Now on to active transport. So like we said, active is using energy. And this is once again still moving across the cell membrane, but this time we're going against the concentration gradient. And there are three types. So we use transport proteins, we have endocytosis and exocytosis. So transport proteins, we've talked about these, carrier proteins. These are going to help pump solutes across that cell membrane. But the reason why it requires energy versus facilitated diffusion that we talked about earlier is because you're moving from low to high concentration, or against the concentration gradient. So we're going uphill when you're using a transport protein with these different things that are moving. Endocytosis, endo meaning enter, is a good way to remember that. So in endocytosis, things are entering the cell. So in this case, the cell's taking in large molecules. It forms some sort of vacuole with a plasma membrane, and then it brings it into the cell. And there's three types we'll talk about. So there's phagocytosis, pinocytosis, and then receptor mediated endocytosis. So phagocytosis is known as cell eating. So cell eating is simply these pseudopods kind of wrap around the food and they pull it in, and then that vacuole forms and brings it actually into the cell's cytoplasm. And this is a great picture down here showing how that works. Pinocytosis then is cell drinking, is what it's referred to as. And this is when just droplets of fluid are taken in, and they're taken in through these tiny vesicles. So as you can see, this down here is a vesicle. It's going to take in that substance, and it fuses once again with the membrane, and it pinches it off and brings it into the cell. And finally, receptor mediated endocytosis. So this is highly specific, as you can imagine. In this situation, the molecule is binding to a specific protein on the cell membrane. And as a result, only those specific molecules can enter the cell. Eventually, you're going to have a vesicle that forms around, and it will pinch off and get carried into the cell like so. So finally, exocytosis. So exo you can think of as exit. So this is essentially endocytosis in reverse. So this is secretion of material out of the cell membrane. So in this situation, you have a vacuole or a vesicle that's inside of the cell. It's going to come up and it's going to fuse with the cell membrane. So once it fuses, and this right here is a great picture of that, once it fuses with the cell membrane, it's going to release those things to the outside, whether it's waste or gas or different particles. They're all going to be released to the outside once that fusion has taken place, because now it's no longer in the cell. Finally, we need to talk about something called cell metabolism. So cell metabolism is basically the way an organism uses what it gets to make energy. So for a cell, it could be getting food, water, nutrients, how it's using those things to make energy. And there's two types. There's anabolic metabolism or anabolism and catabolism. So anabolism is when you have macromolecules that are built from simple molecules. So we're building things up, okay? So this is creating large from small. And this is an example in photosynthesis. So plants are creating and storing glucose through this process. Now the opposite is known as catabolism. So this is when those larger things are broken down into simple compounds. And as a result, this releases energy. So kind of we talked about exothermic and endothermic reactions. This is very similar here. So this would be similar to an exothermic reaction. And for example, cell respiration. So we eat glucose and then our bodies break that down. So that process releases energy that then our bodies use. And this is a good visual here if you want to pause it and look at this picture. It shows food coming in, which is a catabolic pathway, and then it shows macromolecules coming out or being assembled, which is an anabolic pathway. And then it also shows those cellular building blocks that are there in the middle. And that is all for your notes.